Okay, so we're going to go over part one of the nervous system. Here are your objectives. Okay. Um, so the organization of the nervous system, you have the central nervous system, which encompasses the brain and the spinal cord. You have the peripheral nervous system, which encompasses all nerves in the autonomic nervous system. Um, is part of the peripheral nervous system. So we will go over the nervous system that's divided into two parts, central nervous system and peripheral. And then just remember autonomic is part of the peripheral nervous system. So we will be going over all of that in part two. Okay. This is part one today. So we're just going to go over neurons. We're going to go over action potential, synapses, and transmission. So you have the cells of the nervous system, such as the neurons. The neuron structure consists of three main parts: the cell body of the neuron, the dendrites. They are the ones that project, they're branching projections that conduct impulses to the cell body of the neuron. And then the axon is the elongated projection that conducts impulses away from the cell body of the neuron. So the axon is surrounded by a wrapping material called myelin. The myelin is like a slippery layer over the axon that the nerve transmissions go through. And then the nodes of Ranvier are the ones, the gaps between all of those adjacent Schwann cells. So what happens in that case is, say you have multiple sclerosis or a demyelinating disease, what that can do is cause some demyelinization, usually from an autoimmune reaction, where that slippery network does not gets destroyed, and then the transmissions kind of like skip over the junctions, or it's not a smooth transition. And when that happens, we end up with some deficiencies in any nerve transmission or mobility. Okay, so that's what usually happens in demyelinating diseases such as MS. So the thing that you need to know mostly about the neuron is the cell body. The dendrites are the ones that come off of that cell body, and they conduct impulses to cell body of the neuron. And then you have the axon, which conducts the impulses away from the cell body of the neuron. And this is going to become a little more clear. So if you have sensory neurons, they conduct impulses to the spinal cord and brain. So these are sensory ones coming into the spinal cord and brain. They're afferent, means they're arriving. Motor are the efferent neurons. They're conducting impulses away from the brain and spinal cord to the muscles and glands. The interneurons conduct impulses from sensory neurons to motor neurons, or among a network of interneurons, also known as central or connecting neurons. So the axon is surrounded again by that myelin. And what that's doing is transmitting impulses away from the brain and spinal cord to the muscles and glands. And then the nodes of Ranvier, like I told you, are those little spots that are adjacent to the Schwann cells. So here's your neuron. Here's your dendrites. This is what's receiving all this great information. These are the afferents. They're coming in and they're receiving information from the dendrites. You have mitochondria in here. You have a nucleus. And this sends impulses down this axon, going out to either like down the spinal cord into a muscle. So this is the myelin sheath. Here's your Schwann cells that maintain that myelin sheath, and then you have the nodes of Ranvier in between, so this messaging can get transmitted down. So what's an example of this? So we have afferent neurons. So we touch something sharp, it sends an afferent neuron will send an impulse to an interneuron, which will come now to the spinal cord and the brain. It will process that, and it will send an impulse now out to that efferent neuron, which is your axon, and it will be the effector, which means it's now leaving, and that is going efferent, which means exit, right? Efferent is going to send that out to move your finger from that sharp object. Um, the cells of the nervous system. So we have glia. Why are we talking about glia? Glia is the most common types of brain cancer, like gliomas, and that's where they develop from. So glia, though, supports cells. It brings the cells of nervous tissue together structurally and functionality. There's three major types of glial cells of the central nervous system. And they are astrocytes. They're star-shaped, and they anchor the small vessels to the neurons. Then you have the microglia, which are small cells that move in inflamed brain tissue, carrying on phagocytosis. And the oligodendrocytes form myelin sheaths on axons in the central nervous system. The Schwann cells form myelin sheaths on axons of the peripheral nervous system. Okay, and glia vary obviously in size and shape. So here's central glia. These are the central neuroglia. Here you see the capillaries. So these are the ones that are supporting the capillaries. Here's your astrocytes. They have little football. They look like stars, right? Then you have your microglia. So the microglia, here we are. This is just it's a smaller one. And then you have your oligodendrocytes. Here's your oligodendrocyte is here, and it is supporting. So if you remember these three, remember the oligo form the myelin sheaths, the micro have phagocytosis. So what does that mean? Phago means to eat, or eat, phage, right? Means to eat. Cytosis is done by the cell. And then the astrocytes are just anchoring those small blood vessels to those neurons. Okay. So, just keep in mind that our microglia are the ones that are cleaning the messes up. And then your oligodendrocytes have those myelin sheaths. Okay, and there's your nerve fiber. So if you look over here, the peripheral neuroglia, here's your nucleus of your Schwann cell. This is the myelinated nerve fiber. There's your myelin sheath, and this is what's called the neurolemma. Okay. So nerves are bundles of peripheral axons. You have a tract, which is the bundle of the central axons. You have white matter, which is tissue composed primarily of myelinated axons. Then you have gray matter, which is the tissue composed primarily of cell bodies and unmyelinated fibers. The nerve coverings are going to be the fibrous connective tissue. If you remember endo, peri, and epi, we're going to learn this with the heart as well, because the endonorium is usually going to be inside somewhere. When we say somebody's having a root canal, they're having an endodontic procedure, or in the endocardium, if the heart, that is what is lining the chambers of the heart, and the valves. The perinorium surrounds the outside of that fascicle of nerve fibers, and then the epi is surrounds the entire nerve. So if we say epicardium, that's the outer layer of the heart, and what is right in the epicardium is the pericardium. So that's these outer layers, where the endonorium is an inner layer. Okay, so here's your nerve. This is your epinorium, and within that epinorium, you're seeing a perinorium that's wrapping around this big fascicle. Okay, so this is a fascicle, which is a bundle of nerves, and that's your norium. And then what happens in each fascicle, you have these nerve fibers, and this is one nerve fiber coming out with the myelin, and this is in your endonorium surrounding that actual myelin sheath of that axon. Okay, so this just gives you a really good aspect of what nerves are made up of. It's very complicated. When we talk about the signaling, we're talking about like reflex arcs. What that means is a nerve impulse is conducted from a receptor to an effector exiting over neuron pathways or reflex arcs. And that conduction by that reflex results in a reflex, or a contraction in a muscle, or a secretion by a gland, right? So don't forget about that. These nerve impulses can cause secretions from various glands. The simplest reflex arcs are two neuron arcs. They consist of sensory neurons synapsing in a spinal cord with motor neurons, and then you have three neuron arcs, which consist of sensory neurons synapsing in the spinal cord with interneurons that synapse with motor neurons. So this is going to become a little more clear in a second. Nerve impulses are also called action potentials. We are going to talk a lot about action potentials. If all impulse in conduction ceases, then life will cease. Just be aware. So a reflex arc, this is going to give you an example. So we have this reflex hammer. We hit subpatellar area below the kneecap into the patellar ligament, and that will send a sensory neuron or an afferent neuron impulse into the dorsal root ganglion. That dorsal root ganglion comes into the gray matter of the spinal cord. It synapses in there and causes an action potential for something to happen. And what happens is this sends it down the efferent going away neuron, which is your motor neuron. Okay, so efferent exiting the gray matter, going down into the neuron, into the muscle to activate the motor neuron of that muscle through an action potential, which will stimulate the leg to jerk, hence the knee jerk response. So just remember, here's your from the receptor, sensing this stretch receptor, coming into your afferent neuron, the sensory neuron, going into the dorsal horn here, and this is going to be in the gray matter, which is going to send it out through a synapse here, an action potential, and a nerve impulse down the motor neuron axon to the skeletal muscle on the effector nerve, exiting the spinal cord to cause that muscle. The quadriceps muscle is the effector to cause that knee jerk response. So that is the reflex arc in a good diagram. So the definition of a nerve impulse is a self-propagating wave of electrical disturbance that travels along the surface of a neuron. Membrane, also called action potential. We are going to get into this in depth. The mechanism of a nerve impulse at rest, the neuron's membrane is slightly positive on the outside, so it's polarized because you have excessive sodium ions out there. Remember, they're out extracellular, and a stimulus triggers the opening of the sodium channels in the plasma membrane of the neuron, and that causes it to become more positive in the cell. Okay, and we're going to talk about the changes in the polarity. So nerve impulses do not continually race along every nerve cell's surfaces. They are triggered. So the impulse inward movement of that sodium will depolarize the membrane by now making the inside membrane, which was normally negative, more positive than the outside at the stimulated point. This depolarization is a nerve impulse or an action potential, and the stimulated section of the membrane immediately repolarizes. But by that time, the depolarization has already triggered the next section of the membrane to depolarize. So that propagates a wave of electrical disturbances like depolarizations all the way down the membrane. This is going to make more sense in a little more in the lecture. So a synapse is where those impulses are transmitted from one neuron to another. We call that a postsynaptic neuron. The synapse is made up of a knob, a cleft, and a plasma membrane, and neurotransmitters will bind to the specific receptor molecules in the membrane of that postsynaptic neuron, and that will open an ion, therefore stimulating an impulse conduction by that membrane. So the transmission of signals from one neuron to the next is very important role in nerve conduction. Neurotransmitters are very important. I'm sure you've heard some of these. Acetylcholine is an excitatory one that causes muscle contractions and things to happen within the synapse. Catecholamine, such as norepinephrine, is excitatory. Dopamine is actually inhibitory. And then you have serotonin, which makes us feel good. We have endorphins that make us feel good, and enkephalins can transmit pain, nitric oxide, and other compounds such as GABA. So I'm going to talk about a little bit. Think of acetylcholine as being released from a synapse to cause something to happen and an action potential somewhere. So it's excitatory. Dopamine's inhibitory. So here's a good example. I have a patient that has Parkinson. If they have Parkinson disease, what happens is they have a deficiency in the production and the maintenance of dopamine. And we need that because what happens in Parkinson, they have this resting tremor. Okay. If they go to try to pick something up, it usually diminishes a little, but they essential tremors, they tremor all the time. Like it's an intent, like if they go to pick something up, they're going to have it. That's not Parkinson. That's different. But Parkinson has a resting tremor, and then when they go to pick something up, it kind of diminishes. That's how you can tell the difference. But the problem with Parkinson is that they have a insufficiency of dopamine, and they have a misbalance. So acetylcholine is the excitatory, and it's causing that tremor. Dopamine would level that out if they had enough. So what do we give? We give things to inhibit enzymes that break down. We give acetylcholinesterase drugs, which will cause them to keep the acetylcholine a little lower active, and then we can give medications that are like dopamine. We can't give dopamine because it doesn't cross barriers when we take it in medicine. But what we can give is what's called levodopa, but levodopa has a hard time crossing too. So we give a medication called carbidopa levodopa, and that helps get that dopamine where it needs to be to decrease that tremor, because if we give more dopamine, you're inhibiting that tremor. So I'm hoping you understand that like all these neurotransmitters, they're chemicals on how neurons communicate, and they can be excitatory or inhibitory. They have linked dopamine to people that have it's the reward center, they're calling it now, and it's been implicated in addiction, believe it or not. Okay, so let's go over that synapse I was talking about. So if I implode this a little, you see your axon, your nice myelinated axon coming down. You have an action potential that's conducted along that axon. It reaches this knob. Here's your mitochondria. Then the next step is the action potential will trigger the release of neurotransmitters from these vesicles. These vesicles will drop in this, it could be acetylcholine, it could be any neurotransmitter coming over here to another neuron. Then what, or this could be a post-synaptic neuron, or it could be a muscle fiber, and we have acetylcholine. So just remember, whatever's being released here, that is what's being released to cause an effect. So it depends on what neurotransmitter is being released. The neurotransmitter will cross this cleft and bind to the postsynaptic receptors. So things have to happen in order to open that gate. So this neurotransmitter will go back into the plasma of the membrane of the postsynaptic neuron, and then these ion channels will initiate the impulse. Okay, so that's how it works through a nerve ending and how it functions. So let's talk about the central nervous system. Let's talk about the brain. We have the brain stem. We have the cerebellum, which is responsible for balance. When people get pulled over for a DUI, what do? They're testing cerebellum function. They walk the line for balance. They do finger to nose movements. And you have the diencephalon, which encompasses your hypothalamus, the pineal gland, and thalamus. And also, of course, the main part of our brain is this cerebrum. We're also going to talk about the spinal cord and the coverings and fluid spaces. So here's the main regions of the brain. This is a sagittal cut, which means I cut it right down the middle, down my nose, and you'll see the brain split in half. This is your cerebral cortex. This is your thalamus, and here the corpus callosum, and then in here you have the pineal gland, you have the hypothalamus, this is the pituitary gland, and the pons. This is going to be the medulla oblongata, and your midbrain. Okay, so it's medulla oblongata, pons, midbrain, and this is the cerebellum that's responsible for all that good balance. And this is just your cross section of a this was the cross section of preserved brain here. Okay, so the brain stem, what do we need to know about that? It has three parts, and we talked about the medulla oblongata, that's at the bottom, the pons, and then the midbrain is closer up near the thalamus. It's white matter with bits of gray matter, and all three parts of the brain stem will conduct impulses to higher parts of the brain. This is the cerebellum. The cerebellum is the second largest part of the human brain. It has gray matter. Outer layer is thin, but it's very folded. So, like if you took the brain out and spread it out, because it has sulci and these little folds, it would be really big. But we have a small area. So, how we get more surface area is by crumbling it up a little bit and folding it, right? And so that way you have a larger surface for processing information. We also has these internal tree-like network of white matter tracts called arbor vitae. So arbor means tree, vita means life, right? The functions help control muscle contractions to produce coordinated movements, essential for balance and moving slowly and sustaining normal postures. So if someone has a cerebellar lesion, they're unable to do coordinated events or help the cerebrum and other parts of the brain. So a cerebellar lesion, such as with a stroke, can cause problems with balance and mobility and smooth movements and coordination. The diencephalon is the hypothalamus, which consists mainly of, it's small, but it is very important. The hypothalamus is located right below the thalamus, and it consists of the posterior pituitary gland and the pituitary stalk and gray matter. I want you to remember some things about this. We talked a lot about the pituitary gland, the thyroid gland, things like that. Um, and when you think about, we talked about antidiuretic hormone when we were talking about fluid balance. So think of the anterior pituitary. This is a small organ, okay? But it's very important because the hypothalamus is what detects changes, negative feedback, right? Positive feedback. If it sees something high, it'll send a signal from the hypothalamus down to the pituitary gland to say, hey, increase this hormone or decrease this hormone. So that's negative feedback. I want you to picture the pituitary gland as a little Volkswagen Bug, and you have in the anterior, the front seat, anterior pituitary, you have like thyroid stimulating hormone, adrenocorticoid releasing hormone, which sends it down to the adrenal cortex to secrete cortisol. And sex hormones are triggered also by FSH and LH that are triggered in the anterior pituitary. We have prolactin for breastfeeding in the anterior pituitary. The posterior pituitary, if you picture that everybody's in the front seat of this pituitary gland, all these hormones are doing their thing. And then you look in the posterior, you have a pregnant woman who's in labor. So oxytocin, right? And she's holding a fish bowl with water in it. Who regulates the water balance is your antidiuretic hormone. So if you can't remember what's in the anterior pituitary, just remember everybody except the back seat in the posterior is your antidiuretic hormone and your oxytocin. Just picture that pregnant woman contracting, holding a fish bowl of water. The diencephalon also acts as a major center for controlling the autonomic nervous system, so it will control the functioning of most internal organs. The controls, also the hypothalamus controls hormone secretion. This is what I just told you, anterior and posterior pituitary glands. So it indirectly will help control hormone secretion by the other endocrine glands. Contains centers for controlling body temperature, appetite, wakefulness, pleasure, things like that. The thalamus is a dumbbell-shaped mass of gray matter that's in the middle of the brain, and it's towards each cerebral hemisphere. So it relays sensory impulses to the cerebral cortex and sensory areas, and in some way produces the emotions of pleasantness and unpleasantness associated with sensations, and on the arousal mechanism. Oh, sorry. The pineal gland receives sensory information about light and adjusts for melatonin. So just remember the pineal gland um, is what we see when, like, so when we were in Iceland and it was daylight all day, um, we didn't release a lot of melatonin, therefore we felt like we couldn't sleep. So just keep in mind the pineal gland is responsible for that. There's also a lot of information about the thalamus. Like when you learn something, the thalamus will process it and it will determine if it should keep it long term or not. So that's a really good one for learning. Cerebrum, not cerebellum, cerebrum is the largest part of the human brain. The outer layers are gray matter, and that's the cerebral cortex. It's made up of lobes composed mainly of dendrites and cell bodies of neurons. The interior of the cerebrum is composed mainly of white matter. So you have the tracts and the basal nuclei. Okay, so nerve fibers are arranged in bundles. The basal nuclei are islands of gray matter that regulate the automatic movements and posture. And then the functions of the cerebrum are mental processes of all types. That's sensations, consciousness, memory, voluntary control of movements also. And the cerebrum has these ridges or gyri and grooves known as sulci. Okay. So here you'll see this is your central sulcus here. Here's your frontal lobe, your lateral fissure, your parietal lobe, your temporal lobe, your occipital lobe, your cerebellum is down here. So if you look here in the prefrontal association area, this is going to be your conscious thought. In here is premotor. This is muscle coordination. Your precentral gyrus also is somatic motor. This is the central sulcus. And then here behind that is your primary somatic sensory area. So this is body sense perception. Here's your primary taste area. Here's your visual cortex here and your visual association area here. I want you to keep in mind when you hear of a motor speech area, Broca area. So I always, and you have Wernicke, okay? Wernicke is here. This is the sensory speech. Over here is the motor speech. What does this mean? So if someone has a stroke, just picture this brain. Somebody has a stroke in here. Okay. If somebody has a stroke in the front, they may not have very good conscious thought. They may put milk in the mailbox and mail in the refrigerator. Okay. If they have a stroke here, I've heard it changes personality if it's a frontal lobe injury. What about motor coordination? Sensory, we do know some patients that had strokes get what's called self-neglect. So they will not associate that their right arm is their right arm and they just ignore it. And then we have areas for stroke that can happen in the Broca area. And what happens is the patient understands what you're saying, but they can't articulate what they want to articulate. You can give them a whiteboard, they can write it down. I actually had a patient that had what's called Broca's aphasia. He could not speak because he had a stroke that affected the Broca area in the brain. So they can hear it and understand it, but they can't get it out. And I always say when I was in Italy, everything ends with an A. So Broca, my speech is a Broca. I have a stroke there, I can't talk. But I understand. Now, in the Wernicke area, if you have a stroke here, you may not be able to understand what somebody's saying, so it sounds jobbled, so you may speak jobbled, you know. So, think about stroke patients and where the different areas of the brain that we can have issues. And think of one of the most common dementias, obviously, Alzheimer's has now took number one. I think because we can identify it a little quicker now. But vascular dementia was big. So your patients that had atherosclerotic disease or coronary artery disease or, cerebrovascular disease or cardiovascular disease. They get deposits in the vessels, the small ones in the brain, and it can affect and cause them to almost have these dementias because of the anoxia or hypoxia. Okay. Um, so now we're going to the spinal cord. If you are an average height, your spinal cord is usually about 17 to 18 inches long. And if you have some kind of injury that actually damages or cuts the spinal cord all the way across, these impulses can no longer travel up to the brain from any part of the body located below that injury. Keep that in mind. So the spinal cord is columns of white matter composed of bundles of myelinated nerve fibers. They form the outer portion of the H-shaped core of the spinal cord. Bundles of axons are called tracts. So we have interior composed of gray matter made up mainly of neuron, dendrites, and cell bodies. The spinal cord tracts provide two-way conduction paths. You have ascending and descending tracts. The spinal cord functions as the primary center for all spinal cord reflexes. Sensory tracts conduct impulses to the brain, and then the motor tracts conduct impulses from the brain. Remember that, okay? So we get sensory to the brain and motor out of the brain. So this is the spinal cord and the spinal nerves. So if you did a dissection of this, this is what you would see in the C1 to C4. Here's your little spinal, your dorsal roots are here. And then you have your spinal nerves, C2, C3, C4. This is the posterior sulcus of the spinal cord. And these are just the bony transverse process of the vertebrae. And then in between these, you have the foramen. So think about when people get spinal stenosis and they can crush these. And that's what can cause some discomfort and tingling and numbness and sometimes partial paralysis. So think about it, your T, I think more or less T spinal nerves, the upper T's control the diaphragm. So, like C5, C6 can actually have control of your fingers. The lumbar can have control of your muscles in your legs. And then the sacral ones can go all the way down to the toes. And sometimes sexual dysfunction can be related to a sacral nerve issue. Okay. So when we deal with the coverings and fluid spaces of the brain and spinal cord, the coverings are cranial nerves and vertebrae, cerebral and spinal meninges. We have different layers. The dura mater in Latin means tough mother, and that's exactly what it is if you try to peel it off a brain. The pia mater, which is the middle one, and the arachnoid is the outermost. So you have fluid spaces or subarachnoid of the meninges. You have the central canal inside the cord, and then the ventricles in the brain. If you ever heard of somebody that has hydrocephalus, what that means is they have pressure in the brain because the ventricles in the brain are filling with fluid and not draining. Some people have high pressure, some people have low pressure. So if you ever have a patient that you hear they have a VP shunt, be suspicious that maybe they have hydrocephalus, and it's a VP means ventricular peritoneal shunt, which means it drains a lot of that fluid from the brain down into the peritoneum. So it's just keep in mind the ventricles do contain fluid up there. Spinal cord and its coverings, we just talked about this. Here's your spinal nerve, your dorsal root ganglion. This is your gray matter, your white matter. Here's the pia mater. It's that inner area. Then you have your arachnoid mater, which is in the middle. And then your outside layer is going to be this dura mater, this tough mother. It's all meant to protect. Peripheral nervous system. So what we're going to do, we next week are going to talk about the peripheral nervous system. And we're also going to talk about the central nervous system and the autonomic nervous system. So we're going to stop at slide 32 and we will pick slide 32 up when we go over that at another time. Um, what I do want to pull up for you, I'm going to find on YouTube. Um, I want to find a video for you on the nervous system. Let me see. Let me see. She usually has good videos. Let me play this one for you, okay?
Hey everyone, it's Sarah with RegisteredNurseRN.com, and today we're going to go over the neuron. In your anatomy and physiology class, you'll be required to know the structure and the function of the neuron. Behind me, I have a diagram of a neuron that I'm going to go over for you. And then afterwards, I highly encourage you to go to my website, RegisteredNurseRN.com, and take the neuron quiz that goes along with this video and download the study notes as well, so you can be prepared for your A&P lecture exam. So let's get started. The nervous system is made up of nervous tissue. There's two types of nervous tissue. The first type are called neurons. These are excitable nerve cells that transmit electrical signals. The second type are support cells. These are non-excitable cells that wrap around and surround the neuron. The neuron has three specific special characteristics about it. The first is that they have extremely longevity. They last over 100 years. That's why we only have one set of neurons and they last us our whole life. Two, they do not divide. So I'm writing an essay on cats in art history. I'm an art history major, and they can't be replaced. So once a neuron's gone, it's gone. You can't infuse it like you can red blood cells. And thirdly, lastly, they have a high metabolic rate and they require continuous amounts of oxygen and glucose. So they're high maintenance and they use a lot of resources to keep them powered. Now, let's go over the structure of the neuron. So be sure to take some notes so you can write down what we're going to go over. Okay, here we have a basic neuron, and we're going to start with the cell body. The cell body is this whole area right here, and it is sometimes called the soma, and it contains one nucleolus surrounded by cytoplasm. Inside the cell body, you have the nucleus, and the nucleus is a spherical, clear area that contains a dark nucleolus in the center, and I like to remember it as it looks like an eye of an owl, and it's found in the middle. Then you have the chromophilic Nissl bodies, and these are found in the cytoplasm and continually renew the membrane of the cell and the protein part of the cytol. And remember in our other video when we went over the cell, the cytol is just the liquid found inside the cell. And then next, you have some lysosomes. And again, we remember from that lecture that these just digest free radicals or any germs around. I like to remember that by the product Lysol. We spray Lysol disinfectant to get rid of germs. So that's all that little thing does. And then next, we have the mitochondria. And the mitochondria, of course, is the powerhouse of the cell body. It keeps everything powered. And then next, we have neutrofibrils, and these are these little pink strands throughout this cell body. And what this is a bundle of intermittent filaments that run in a network between these chromatophilic bodies, and they keep the cell from being pulled apart. So it just keeps the structure nice and sound. And then next, we have our dendrite. The dendrite, as you can tell, looks like limbs on a tree. So whenever you're having to label your diagram, remember these little limbs on a tree, and that's your dendrites. And your dendrites are receptive sites that conduct electricity toward the cell body. So they direct the signal toward the cell body. So always remember that. That's usually a test question. Test question usually says, does the dendrite conducts electrical signal blank towards the cell body? So it would be towards instead of away, which we'll go over axon here in a second. And then you have your initial segment. The initial segment arises from this area, which is the axon hillock, which helps the neuron to split apart and branch down through your axon. So that's just a little part, and it's this area right in here. And then the axon hillock actually means a little hill, and this is just where it's moving up in a little hill formation, and that's just this area right there. And next we have our axon collateral, and this is where this axon is actually splitting off. And this is where it just extends the axon at right angles. So whenever you see it splitting, that's where your axon collateral is. Now to the axon. The axon conducts electrical signals away from the neuron. So remember, axon starts with A, and it will take the signals away from the neuron. Now remember, the dendrite took it toward. I like to remember dendrite starts with D, it drags it towards it. But the axon carries it away. That's usually always a test question. So remember that about axon. Next, your terminal branches. This is where your axon has branched off and then it's forming the axon terminals. This is just where you have tens of thousands of these per neuron, these little branches, which is absolutely impressive. And it just helps form the axon terminal. And the axon terminal are these little knob formations, and this is where they contact each other. They contact other neurons to form synapses. And then last but not least are your Schwann cells, and these form myelin sheaths around the axons. So that's all that those little blue things do. Now let's go over a couple quiz questions. Let's see how well you grasp the material of the neuron. Now let's go over our first quiz question. Which of the following is not a characteristic of a neuron? Is it A, they do not divide, B, they have longevity, C, they originate in the embryonic pharynx, or D, they have a high metabolic rate? Remember at the beginning of the video, I went over 3 special characteristics of a neuron. The first one was that they have a high metabolism. The second was that they have a longevity, meaning that they last us their whole life, 100 years. And then 3, they do not divide. They can never be replaced. So those are the 3 special characteristics of a neuron. So the answer to this question would be C, that they do not originate in the embryonic pharynx. That is not a special characteristic of it. So our answer is C. Now let's take our next quiz question. Let's see what the question says. This is called the soma of the neuron and has a single nucleus that is surrounded by cytoplasm. So let's think back to that drawing we had on the board. And let's see what our options are. A, Nissl bodies, B, cell body, C, ganglion, or D, neutrofibrils. So what are Nissl bodies? Remember on the diagram that I had that those were the green little circles that were inside the cell body. What Nissl bodies do are they renew the cell membrane. So we know that it's not that because we're looking for something that has a single nucleus in it and it's surrounded by cytoplasm, and it's also called the soma. So let's look at the next option. The cell body. That is definitely the answer. The cell body is also called the soma, and it was the whole area where everything was inside of it, which are your Nissl bodies, your nucleus, your lysosomes, your mitochondria. The cell body contains all of that. So the answer is cell body. But let's see why it wasn't C or D. It wasn't C, ganglion. We didn't even go over that term. That was just something in there to throw you off. And D, the neutrofibrils. Remember, the neutrofibrils were those pink little strands within the cell body, and that was just to help keep the cell from collapsing, keep it strong. So that was the answer to that. Now, take rest of the quiz on my website, registerednursern.com. A link should pop up so you can take that.
Okay. So I hope that gave you a little bit of a review on the neuron, just so you get that. And then what we're going to do, we're going to go over this PowerPoint about action potentials. And then I'm going to show you a video about action potentials. I stole this PowerPoint. I have SlideShare subscription, so I was able to get it off there. this is a good, it's a really good step-by-step PowerPoint. So we're going to talk about the 5 steps of action potentials. This is going to become very important when you learn your antiarrhythmics in cardiac pharmacology. It's going to help you a ton to understand all about electrophysiology. So there's 5 steps of an action potential. We have a resting membrane potential. This occurs in different cells called excitable cells, which include the neurons, the muscle cells, the endocrine cells. There's a resting membrane potential. You have the threshold, which is early depolarization. We have depolarization, repolarization, and hyperpolarization, where you have this undershoot and refractory period. That's due to the sodium potassium pump. So we're going to go into this. So positive or negatively charged electrolyte can cause a difference in the electrical charge between intracellular and extracellular portion of a neuron. That is called a membrane potential. So when you look, the nerve impulse this nerve impulse can actually influence the movement of electrically charged ions across the cell membrane. So what does that mean? During a resting state of a neuron, the potassium concentration is higher inside the cell. If you remember, we talked about all this, who lives in the cell, and who lives outside the cell. And if you remember, potassium lives inside the cell, right? And it lives, so you have the potassium inside the cell and the sodium outside. And the sodium and chloride concentration is usually higher outside the membrane, because remember, opposites attract. So you have sodium chloride, and chloride stays on that membrane. And the resting membrane potential is about -70. So chloride's important in that concept, that we do like to keep the resting potential at about -70. Okay. Um, so then the other thing that you want to keep in mind is the resting membrane is about -70, but the neuron overall is -70, but the potassium ion concentration is higher inside the cell. And then you have potassium will want to leave the neuron if it can. Okay, she loves her little space in there, but um, she can, leave if she can. So then the sodium ion concentration is higher outside of the neuron. So the sodium will want to enter the neuron if it can. But the channels are usually closed, so the ions can't really diffuse. So early depolarization is called threshold. And the action potential starts by a stimulus causing that resting potential from -70 to going to 0. What does that mean? We're going more positive, right? So now when depolarization reaches about -55 millivolts, a neuron will fire an action potential, and this is the threshold. Okay, so typically our membrane depolarization threshold is about -55. So it goes a little more positive. If the neuron does not reach this critical threshold, then no action potential will start. And in this step, some sodium channels open, which causes sodium to move through the channel into the cell due to some stimulus. And remember, sodium's a positive charge. So the inside of the neuron becomes more positive and becomes depolarized. Outside of the neuron becomes negative. So after we reach threshold level, more sodium channels open, which causes this huge sodium to move through the channel into the cell. So here's outside the cell. We're negative outside now. We're going in, and now we're positive. So this is depolarized. So the inside of the neuron becomes more positive, and this is what we refer to as depolarized. So the membrane potential will reach from -55 millivolt to +40. Okay, and this is the membrane potential. After depolarization, you get this huge sodium inside the neuron, and membrane potential will reach +40. And then the sodium channel inactivated and sodium channel open. So the sodium channel now becomes a little inactivated, okay? And we are now what? We're negative inside the cell, positive outside. So what has happened? The outside of the neuron becomes more positive and becomes repolarized because the potassium channel opened, and that caused the potassium to go outside. Now we're real positive again on the outside. Chloride will move from the outside to the inside to turn this in a little negative. Okay. And the membrane potential falls now because this is repolarization, right? Because depolarization is more positive. Repolarization is going to go to -70. So now during hyperpolarization, so now we're even going to go more negative. So the voltage potassium channel stays open just a little longer than needed to bring the membrane back to its resting potential, but sodium channels are now closed. This is called overshoot. So the membrane potential becomes more negative, almost to -75 millivolts, than its resting potential. After that, the potassium channel will close and normal potassium can move from inside to outside. What is the benefit to this? Let me tell you, because you have this slow closure of that voltage gated potassium channel, you get that undershoot, which contributes to a refractory period. What does that mean? It makes it harder to depolarize the membrane. Even once the voltage gated sodium channel has returned to its active state, we need refractory periods. When you learn about arrhythmias and you learn about antiarrhythmic therapy, you're going to learn that we want to try to prolong the refractory period a little bit, because what happens when during refractory, both sodium and potassium channels are closed, and it ensures that another action potential will only travel forward down the axon, not backward, okay? So during the refractory, you have a sodium potassium ATPase pump. They get activated by an ATP molecule. That pump will allow 2 potassium to go back into the cell in exchange for 3 sodiums to come outside, and that is what will make that resting membrane more at a resting membrane potential. So what's the deal with this? So when the action potential reaches the axon terminal nerve ending, it causes a neurotransmitter containing vesicle to fuse with that membrane, releasing the molecules into the cleft. That was that space that I showed you earlier. And then what it does from there, it actually will cause neurotransmitters to bind to this ligated ion channel when receiving the cell, and it may undergo depolarization and undergo its own action potential. So this is what's happening. And I want to just stress to you, the refractory period is very important. We have what's called a relative refractory period. That means that a higher than normal stimulus will cause an action potential. And if you have an absolute refractory period, that's the period where no impulse can get through. So this is really protective of having arrhythmias. So when you're going to know this schematica, except up here you have the sodium and potassium, we're going to have calcium involved in here when we start learning about this in pharmacology. But I just want you to keep in mind, we start out at -70, and what happens? The sodium channels open, and that makes it go here to -55, a little more positive. And what happens there? The sodium channels are open, more of them are open, and we're going to have an action potential and move that measurement more positive. So then once we hit depolarization, we're going to move over. You will see calcium channels will be involved here, but we're not going to talk about that here. Then in 4, repolarization, you're going to have a sodium channel close. You're going to have the potassium channel open, so a little bit more potassium goes out. Then it comes to a point where now we're in repolarization going to hyperpolarization. And at that point, your potassium channels will close. And then we'll have that channel where the sodium, you get 3, the potassiums will exchange the sodium, okay? So they'll exchange, and that is what's going to bring it right back to its resting membrane potential. But you're going to have that hyperpolarization where we have those refractory periods. Okay, so again, here's another figure showing the first thing is a resting state. Your most sodium's outside, your potassium's inside. The inside membrane in the cell is negative. And what happens is you get an action potential and depolarization happens where the sodium comes into the cell. And now we have a depolarization. And then in repolarization, we start to get these, the sodium will go back and we'll have that positiveness on the outside and we will go back negative on the inner membrane of the cell, and then you'll get some potassiums continued out, but then we get the sodium potassium pump and it will normalize after undershooting and back to resting. So this is another just showing where it's, resting, we're negative inside. And then what happens is depolarization will spread as sodium rushes into that cell to make that inner cell membrane more positive. And then you'll have the exchanges. And this is what's happening during the axon. Okay, so that just gives you some information there. I kind of wanted to go over, like show you a video of this action potential, and I'm going to try to find that for you. Okay. Let me go on YouTube and get you a little schematica here. Okay. Action potential. YouTube. See if I can find the one that I like. Is it Let me go to YouTube, but let me
What of everything you
sorry, I want to find the there's one that I like and I can't find it. Let me do this one and see how long it is.
Hi viewers, welcome to Pharmacy D by Asim. This is Dr. Mahnoor Khan, and in this video I'm going to explain an action potential. But first, if you haven't subscribed to my channel yet, please subscribe Pharmacy D by Asim and keep following us for informative videos. Moving towards my topic, that is action potential. In simple words, action potentials are nerve signals. Neurons generate and conduct these signals along their processes in order to transmit them to the target tissues. An action potential or nerve impulse causes a movement of ions across the cell membrane of a neuron, similar to a ripple passing along the surface of a pond. The cell membrane of the neuron contains thousands of tiny molecules known as channels. These channels allow either sodium or potassium ions to pass through the membrane. Generally, the channels on the neuron are closed and the membrane is said to be in a resting state. In this state, the charge of the inside of the cell membrane is more negative than the outside. The voltage inside this membrane is about negative 70 millivolts, meaning that the inside of the neuron is about 70 millivolts less than the outside. The cell membrane of a neuron is polarized because of this electrical difference across the membrane. Now, a nerve impulse starts. When pressure or other sensory input disturb a neuron's plasma membrane to the point that the potential difference reaches a threshold voltage of about negative 55 millivolts, this causes hundreds of sodium channels in that region of the membrane to open briefly. This in turn allows positively charged sodium ions to flow inside the cell membrane. Now, the inside of the membrane temporarily becomes more positive than the outside, which in turn causes the voltage to rise. This is called depolarized state. The membrane is now depolarized because the net charge inside the axon changed from negative to positive as sodium ions enter it. As the impulse passes, the potassium channels begin to open, allowing positively charged potassium ions to flow out of the membrane, which cause inside of the axon to resume a net negative charge. At this point, the voltage would fall. The membrane is now said to be repolarized because again it is negatively charged on the inside and positively charged on the outside. So, this depolarization and repolarization of a neuron membrane is called an action potential. Action potential is just another name for a nerve impulse. After a nerve impulse is some voltage gated potassium channels remain open, resulting in further movement of potassium out of the cell. As the voltage falls below the reading of the resting state, the membrane is said to be hyperpolarized because it is even more negative than at the beginning. At this stage, neuron is unable to conduct a nerve impulse and it's said to be in the refractory period. The refractory period is a very short period during which the sodium potassium pump continues to return sodium ions to the outside and potassium ions to the inside of the axon, thus returning the neuron to resting potential. So, in summary, an action potential is just a wave of depolarization and repolarization. It's not an electric current, it's just a series of voltage gated ion channels open and close. So, viewers, that's all for today. Thanks for watching. Make sure to like and share, and hopefully, I made it really easy to understand. Thank you.
Okay. So, just try to keep all of this in the back of your mind about, keeping these in your mind when you take cardiac pharm. So, that's it for today. I hope you got a lot out of the voiceovers, and we will see each other for part 2 of Neuro, where we will go over the peripheral nervous system, central nervous system, and autonomic nervous system. Thanks.